Hello all. Today's topic is about nucleic acid and enzyme. And I'm going to talk on nucleic acid and enzyme. So first is nucleic acid. Nucleic acid are long chain polymeric molecules, and the monomer is the repeating structure of this nucleic acid is nucleotide, and hence sometimes nucleic acid is also called as polynucleotides. And nucleic acid, there are two types of nucleic acid: deoxyribonucleic acid, that is DNA, and ribonucleic acid, that is RNA. These two are the major type of nucleic acid, and these two are responsible for inheritance and transmittance of the specific characteristic from one generation to the another generation. And these deoxynucleotides and RNA, I'm going to elaborate on. So first is DNA, that is deoxyribonucleic acid. Deoxyribonucleic acid composed of pentose sugar, phosphoric acid, and some cyclic chain or cyclic bases containing nitrogen. So sugar mostly present in the DNA molecule is beta D2 deoxyribose, and the cyclic bases that are present are adenine, thymine, guanine, and cytosine. These bases and their arrangement play very important role in the DNA for the storage of information from one generation to the next generation. And this DNA has a double-stranded helix structure that is complementary to each other. Then comes an RNA. RNA is ribonucleic acid. This ribonucleic acid also consists of phosphoric acid, pentose sugar, and cyclic bases with nitrogen. So RNA has beta D ribose sugar, and they contain adenine, thymine. They contain adenine, guanine, uracil, and cytosine as their nitrogenous bases. This RNA, uh, this fourth base, that is uracil, is different from the DNA in the RNA. So RNA generally consists of single strand that sometimes fold back and make a double strand. These RNA are of three types: mRNA, rRNA, tRNA. M means messenger RNA, R means ribosomal RNA, and T means transfer RNA. Transfer RNA. So this is all about RNA and DNA, that is nucleic acid. And the function of nucleic acid is, very first, they are responsible for transmission of inheritance character or person's identity from one parents to the offspring, means from one generation to the next generation. Then they are also responsible for production, also synthesis of protein in our body. Then they play a very important role in identification of particular persons by their genetic information. So DNA fingerprinting is a technique that is mostly used in forensic sciences, and also it is help to determine the it is help to identify the criminal as well, also to check the paternity, and it play a major role in studies regarding biological evolution and genetics. So this is all about DNA and RNA, that is nucleic acid. Now comes an enzyme. Enzyme, human body composed of different types of cells, tissue, and other complex organs. So for efficient functioning, our body releases some chemicals to accelerate the biological processes. These biological processes include respiration, digestion, excretion, and many more. Since this enzyme play very important role or pivotal role in living entities that governs these biological processes. So what are enzymes? Enzymes can be defined as a biomolecules or biological polymers that catalyze biochemical reaction. Majority of the enzyme are protein with some catalytical properties that are crucial in different processes. So metabolic processes and other chemical reaction in cell are carried out by this set of enzymes. So enzyme play very important role in accelerating the process of this metabolic reaction. So structure of enzyme. Enzymes are a linear chain of amino acid that give rise to three-dimensional structure. This sequence of amino acid are specific for the structure that identifies the catalytic activities of the enzyme. So upon heating, this enzyme can denature, so they ultimately lose their enzymatic activity. Therefore, this is typically these enzymes are some that are typically associated with the temperature. So classification of enzyme are classified into 6 types: oxidoreductase enzyme, transferase enzyme, hydrolysis enzyme, lysis enzyme, isomerase enzyme, and ligase enzyme. So the oxidoreductase enzyme, as the name suggests, oxidoreductase, that catalyze the oxidation reaction where electrons tends to travel from one form of molecule to the another. Then comes transferases, as the name says, transferase, they transfer. So this enzyme help in transportation of functional group among acceptor and donor molecule. Then comes the hydrolysis. Hydrolysis added water, so they add water and make a hydrolysis reaction to cleave the bond and hydrolyze it. Then comes the lysis. Lysis also adds water, carbon dioxide, ammonia across the double bond, and they eliminate these to create some double bonds. Then isomerase enzyme catalyze the structural shift that present in the molecule. So they help in changing the shape of the molecule. Then comes the ligases. Ligases is the enzyme also known as to charge the catalysis of ligation process. So this is all about enzyme and their importance, or their importance in some processes. Then comes the cofactor. It is non-proteinaceous substance that is required for enzyme, that associated with the enzyme, and these are essential for functioning of enzymes. So enzyme have particular characteristic. Enzymes speed up the reaction by lowering activation of energy of the reaction, and this process does not affect the nature and properties of the end product. So enzyme does not take part in the processes, but they just accelerate the reaction, and they are highly specific. So this enzyme catalyze one kind of substrate. Small amount of enzyme can accelerate chemical reaction as well. So enzyme are sensitive. They are sensitive to change in the pH, temperature, and substrate concentration. And then turnover number is defined as the number of substrate molecule transfer per minute by one enzyme molecule. So these are all characteristic of enzyme. So this is all about enzyme and nucleic acid. Thank you.